information. So thinking about the soil environment, things like pH, salinity levels, soil temperature, water content, and aeration, what food or nutrient sources are available to pathogens and other organisms, and then what other organisms are in the soil that might interact with the pathogen. And so, again, these are ones that make up a big part of the things we consider with soil health. There are also things that are very dynamic and can be sometimes hard to control in the field. So it's one reason, all of these factors influencing disease incidence is a big reason why sometimes in studies that are done in the lab or the greenhouse in very controlled settings, we might see impacts of disease suppression that we don't see in the field where these factors might be more dynamic or harder to control. So I want to talk a little bit about kind of these theories of suppression and different types of suppression as well. So there's a continuum of general to specific suppression. And general suppression is really kind of the idea that the overall microbiome and its activity is inhibiting pathogen growth and activity. So it could be potentially broad spectrum relating to a variety of soil-borne diseases. It tends to be enhanced by practices that increase the microbiome community size, activity, and or diversity. And it's been shown that increasing soil organic matter may increase general disease suppression. And so this is where, again, where soil health management comes in, is when we think about soil health management, we are often thinking about practices that will also, increase microbial activity, size, and diversity. And so in theory, this would also increase general soil-borne disease suppression. And soil-borne disease suppression tends to be reduced by soil sterilization because you're kind of knocking out a lot of the microbial community. And so how this works, the idea is that a lot of pathogens are in the soil in dormant forms, and so they're responding to root exudates, sugars and amino acids and other nutrients that are in that root rhizosphere area. And so when they detect those, then they might germinate and start to become active. But if the general microbial community is basically consuming all of those sugars and amino acids coming out of the roots, then the pathogens may not be able to detect them as readily. So it can kind of help suppress germination of these pathogens. So that's kind of the concept behind it. Specific suppression, on the other hand, is the direct attack of a pathogen by another organism. And a big aspect of this is that it can be transferable between soils with inoculum. And so I'm going to talk a little bit about some examples of this, but it's really about the population of a specific organism rather than an overall community. So maybe less affected by nutrient additions or organic matter additions, and more affected by the presence of a particular organism. And one thing to note is that this can tend to come up when one plant is continuously, one host is continuously in a soil, so in monocultures, continuous monocultures. And it tends to be kind of an up and down cycle. So there might be disease that comes up because of continuous monoculture. That then makes the antagonistic organism increase its population. It decreases the pathogen, and there's disease suppression, and then the cycle goes up and down again. Whereas with general suppression, it tends to be maybe more stable because there's just kind of this general continued activity of the microbiome. But I want to emphasize that this is a continuum, and so it's important to think about what mechanisms along this continuum are at play with disease suppression in raspberry systems. Again, these are some mechanisms. I've talked about some of these, but just getting into it more specifically, on the specific suppression side, you can have bacterial attachment to pathogens. You can have bacteria or other organisms producing toxins or antibiotics that directly affect the pathogen. They can produce volatile organic compounds, or they can directly destroy the cells, maybe through cell lysis. And then on the general side of things, again, there's this competition for resources between quote unquote beneficial or just the general microbiome organisms and and the pathogen, competition for root infection sites, limitation of resources for spore germination. And then there can also be induction of plant defense, which is also an important mechanism of suppression. So I'm just going to briefly talk about some examples. There's been, on the general side, there's kind of a classic example of Phytophthora cinnamomi root rot in Australian avocado orchards that was shown to suppress Phytophthora root rot. And so this is in a rainforest situation, so very wet climate that tends to favor this host. But in areas where the rainforest had been cleared and avocado orchards had been planted, there was a lot of soil deterioration, soil acidification, loss of soil organic matter, and that brought up extensive root rot. But there was an orchard that maintained soil organic matter levels similar to the native rainforest, so very high at 12%, which is, we often don't see soils that high around here. But they brought in a lot of additions of poultry manure, crop residues, calcium, maintaining that pH at a more neutral level, and they did not have any root rot issues. And this, the thought behind this was that there was just, by maintaining this high level of soil health and soil organic matter, they were feeding the microbes that could then suppress the Phytophthora root rot. On the other side of things, on the specific side, a classic example is take-all disease, which happens in wheat. And there's been shown to be what's called take-all decline occurring with continuous monoculture of wheat or barley, because antibiotic-producing Pseudomonas grow and in response to increased levels of the pathogen. And it's something that has been shown to be transferable between fields, meaning you could take a little bit of soil from a suppressive field, move it to another field, and then see that there's suppression forming in that other field. So again, these are two examples along this kind of on either end of this spectrum. And as we move into the next few talks, some kind of leading questions I want to pose that we need to think about in raspberry systems are, which mechanisms are more at play? Is there more general or specific suppression going on for major pathogens and pests in red raspberry? What cultural practices enhance microbial activity and diversity and general suppression in red raspberry systems? And then also thinking about what cultural practices might exacerbate pathogens. There's a lot of focus on, maybe adding composts or adding green manures that might, could feed the microbiome and could lead to suppression, and also it can lead to, feeding pathogens and pests. So there can be issues with exacerbation that we need to consider. How are soil microbial communities functioning together to suppress pathogens? There's a lot of new tools we have to really understand the microbiome, but there's a lot we still don't understand about how different organisms are interacting with one another. So I think this is an area of research going forward that we really need to understand more and have soil scientists and pathologists and microbiologists all work together. And then also, of course, there's physical and chemical aspects that are important as well, things like soil aeration, pH that might need to be addressed that can be contributing to pathogen issues, not just the biology. So I'm sure that the next few talks will help answer some of these questions, and then we'll have a panel to then talk about what we still don't know and what questions we need to really think about going forward. So with that, I'm happy to take any questions or comments, or we can move on to the next speaker as well. Molly's giving me this signal.
I think we'll take questions. For any questions that you have for Deirdre, you can bring into the panel and she can answer them there just so that we can stay on time.
Thanks.
Thank you, Deirdre. Next up will be Mark Bolda, who is a farm advisor, and he will be talking about soil-borne pathogen management in California, a case study.
Cool. Is Mark in here? I just saw him. Okay, well, that gives me extra time to pull this up, I guess. Mark, are you in the building? He's coming. Okay, great. Yeah, big entrance. Yeah. Okay. No, no, no. I know. That's why I was like, he was just here. He was just here. Okay, and here is Mark.
Okay, very good. Is it, so I don't have a pointer with my,
You don't.
Okay.
You just use those keys there.
okay, very good. Good morning, everybody. Mark Bolda. I think now you know I'm the guy that came in late. Sorry about that. I hope I didn't keep you waiting. You also noticed my sister and my mother are sitting right here. So you're going to get a really good show. I'm going to do my best, right? Okay. So anyway, again, thank you, Chris, for inviting me this morning to come and speak about red raspberries in California. I think there's some things I think that you'll find interesting. What we're doing in California, it's a bit different, but there's always something to be learned when we share. So again, Mark Bolda, UC Cooperative Extension, and I just came in yesterday. So as I always do, I like to just do an introduction. Just so you know, it's about 20 minutes long, the talk. And I've been given a half an hour. So if you have questions or anything, please raise your hand and we'll take it from there. Okay. We have a lot of time to make this a conversation so you can really learn. So first of all, I'll do a short description of California red raspberry production. Then I'll do a description of pathogen. There's a second one. I think that's going to be worth some treatment. I will also be speaking about the AgDia immunostrips. That's a new technology for diagnostics in the field that I think you'll find interesting. And then we will get into talking about soil fumigation and then some of the areas that we have been working on as far as alternatives. Lastly, I know Chris was, he was going to have a separate section on labor that got taken away, but I do have a final slide. I'm going to make some comments on labor situation because it has, as you know, it's becoming rather interesting for us in horticulture with labor. So just in the way of introduction, the California red raspberry industry, it's different than here because it's all fresh market. There's no processing. We don't do juice. We don't do frozen. It's all fresh market, all fresh picked. It's a total of about 4,600 acres or so. About 1,600 acres are up in the Watsonville Salinas area where I work. That's about an hour south of San Francisco. Then we have the Santa Maria area. That's in Santa Barbara County. It's about 650 acres. And then the Oxnard Plain, which is a surprising amount, about 2,600 acres. Again, I underline it's all fresh market. Interestingly, last week I was reading an article in the Wall Street Journal about the red raspberry industry. 60% of our raspberries that are consumed in the United States aren't from the United States. They come into us from Mexico, both frozen and fresh. That's changed from what it was. It was around 30% in 2012 and it's jumping up. So that part of the market is changing quite rapidly. And lots of people come down to California, seen our industry a little bit. Okay, so you see all the tunnels, right? About 90% of our industry now, we grow them under what are known as these plastic tunnels. They're not greenhouses. They're simply structures that are raised and then there's a thick plastic that's put over them. So this started really in the late 90s. There was some research that was done in Portugal and it took off like crazy in California. There's really two advantages to using the plastic. It's expensive. It's about $10,000 an acre to run this thing and maintain it. But there's two advantages. The first, I think a lot of you know red raspberries, they grow in forest clearings, very diffuse filtered light. They don't like direct sunlight, right? So that's good. They perform better. The growers refer to that as being the tunnel magic. But the other part of that is, in California, I think most of you know it doesn't rain in the summertime, but still we do get rain in May and June. And if you have a full crop of raspberries on in May and June and you're not covered and you get rain on top of that, you're going to lose your entire crop, right? I mean, raspberries isn't a crop that goes 6 months. It's those 4 to 6 weeks and that's all you have. You get a rain on top of that, you lose your entire crop. All right. And beyond that, for the buyer, it makes a lot of sense to be working with people who are under a tunnel because that way, if there's a rain, they don't lose their, what they were supposed to sell. If you're Walmart and you're sending out the flyers, raspberry sale and everything, and it rains also, you don't have raspberries, not a good look. So the tunnels matter, and that's why we have 90% of our industry underneath the tunnels right now. So I realize this isn't going to be the first time you've heard about Phytophthora, but I will just cover a few basics about it so we kind of are on the same page. So Phytophthora is the main pathogen. As you know, there's many different species. I know Deirdre was mentioning Cinnamoni. The aggressive one that we have in California is Cactorum. We also have Phytophthora fragaria, many different kinds. It really is the soil pathogen that we deal with when we're talking about red raspberry in California. The picture on your left, that is actually Phytophthora, which came in a nursery stock. And then the picture on the right, you can see that those smaller plants are having difficulty establishing because Phytophthora consumes the roots and it's not allowing that plant to grow properly. And what happens is, you all know this, fruit is what, 90% water? And so if you have a full-grown plant, also it needs a lot of water, its roots are compromised because the Phytophthora is eating the roots. You get these things, you see there's a strip right there where the plants are dead, dying. It's Phytophthora in the middle of the season. Let's talk a little bit about the biology. Again, many of my colleagues here in Washington have done a better job on this, but again, I just want to get into some of the basics because I'm going to dig in a little deeper. One is that Phytophthora actually isn't a fungus. It's defined as being an algae. And when you think algae, what do you think? You think about water. So when we're talking about Phytophthora, where are you going to find Phytophthora? You're going to find Phytophthora where it's moist, where you have a lot of standing water, where the soil is heavy, where you have low spaces. And why does this matter? Because if you look at the picture on your right-hand side there, you'll see they're called the sporangium. It holds all the spores of the Phytophthora. But the picture that I want you to really look at is the one on the left. Those are spores of Phytophthora. They're called zoospores. So when you go to the zoo, you're looking at animals. Animals move around, and zoospores also move around. They have little tails, and they swim through the water like tadpoles. So if you don't have water in your field that's in excess, they're not going to be able to move around. So zoospores are important. So again, that just underlines how important it is to not have excess water in the field when you're growing red raspberries. They're very sensitive to Phytophthora. One other note, I did say I was going to speak about other, I guess I don't have it in here. Oh, that's right. It comes later. Let me talk about a diagnostic technique. Is anybody familiar with these AgDia immunostrips? Oh, this is great. This is really great. I'm going to make a difference this morning, because just listen to me. How much does your soil sample cost when you send it in the lab? $150? Am I about right? It's about right? Okay. These strips, there's 25 of them. You see that little bottle in the middle of the picture? There's 25 little strips in there, and you can do 25 samples for Phytophthora, okay? And these are a lot like, everybody knows this, most people have taken COVID tests now, right? I just took COVID test 2 weeks ago, right? You swirl it around your nostril, and then you extract it, and then you watch on that little cassette that you got your control line and you have your treated, okay? And you're going to do the same thing with these AgDia strips. You bring in the sample from the field, and you can see down the left that you have those roots, right? And you mash up the roots, you macerate them, right? And then you stick them into that bag of buffer solution. The buffer solution kind of extracts whatever in there. And then on the side, you'll see on the side, you see that strip that's been inserted in there? There's 2 lines on that strip. The top line, just like with your COVID test, is a control line that says the liquid is actually moving up. And the second line is what tells you that that is Phytophthora. Okay? And it works. I got a test here. You got all 5 bags. If you look at those close, every single one of them has a line in there. So why do I sound so confident about these AgDia immunostrips? Because over the past 3 years, what I did is test at the same time that a test went into a regular, good diagnostic lab, I did my AgDia strips. And if it came back Phytophthora from the lab and my AgDia immunostrip also said Phytophthora, that's good. And you remember in California in 2023, we had a lot of flooding. So it was a great time for Phytophthora testing. And it worked every time. I'll tell you, I had a case. It was perfect for Phytophthora. I kid you not. It was flooded, it was wet, and it was early in the season. Took the sample, sent the sample into the diagnostic lab. Sample from the diagnostic lab comes in for Fusarium. It wasn't Phytophthora. And you know what my AgDia strip said? 5 different strips, so that's and all negative. No Phytophthora. Okay? They work. And I know Driscoll's has been working with them too. I talked to them. So you have 25 strips for $150 and you can do the diagnostics in the field. You'll have the results within an hour, just like you do with COVID. People are asking then, so it just gives you Phytophthora, it doesn't give you the species. But the thing is, in the field, to make a diagnosis, you don't need the species to take action, right? Phytophthora is good enough for you to make your decision. So this is actually a very good tool for growers for field diagnosis. I highly recommend it. I've been doing this now 3 years, and every single time it works, and our industry partners at Driscoll's are doing the same. I do want to mention, we do have a second pathogen, and this is Fusarium oxysprum form species Fragaria. Without getting into all the details of this form species thing, but this is a Fusarium that we have in California, which is specific to strawberry. It doesn't kill anything else. It does, however, and that's why I'm talking about it at a red raspberry conference, it does, however, grow on red raspberry. It thrives on red raspberry without causing disease so far. So keep your eye on this. And also, I make that note on the bottom too, for strawberry growers, we do have Fusarium oxysprum form species Fragaria race 2. And that race actually overcomes the resistant genetics that we have in strawberry. So all I'm saying is that this pathogen moves around a lot genetically and could be showing up on our raspberries. So pay attention to what's going on. That's why I bring it up. I have the attention of many people here, and I thought I would share that. So are there any questions on the Phytophthora or the Fusarium, the biology idea strips? Yes, sir. Okay, so if I get your question right, you're asking, is there one strip for Phytophthora and then there's another one for Fusarium? It's only for Phytophthora, not for Fusarium. And I will, since now you opened the door for me to expound a little bit further on Fusarium oxysprum. There's thousands of genera of Fusarium. And what happens is sometimes we see diagnostic labs send back, well, you have Fusarium. And people are like, oh my God. No, but the thing is, you need to get it down to the form species. Otherwise, you don't know what you're talking about. There's actually Fusaria which are beneficial. So there's no strips for the Fusarium, unfortunately. When there are, maybe Chris can invite me and I can talk about it a little bit more. Yeah, thank you for the question. Okay, so let's talk a little bit about disease management. Again, this is something that all of you know quite well. You do have the fungicides Ridamil and Aliette. As I always emphasize to our growers in California, the pre-harvest intervals for these are very long. Ridamil is 45 days and then Aliette is 60. Ridamil is very expensive, but it is effective. And then we get to the fumigants. You're still fumigating here, right? Fumigants are still, okay. So this is becoming, I was just up at the California EPA 2 weeks ago, and man, they don't want those fumigants. They are really down on these fumigants. Hundreds of pounds of material per acre of a volatile, toxic gas. We've got to come up with something. And they're like, hey, you get to work. We've been working on this for a long time, and I will share with you some of the alternatives we have going, okay? So, but the fumigants are still what we are using for our red raspberries. There's a huge effect of using the fumigants that benefits the plants. So let's talk about the alternatives right now. So this one you're familiar with. I'll just give a summary of it. This is anaerobic soil disinfestation. The principle of anaerobic soil disinfestation is very simple. You change the environment of the soil by taking away the oxygen temporarily, right? Anaerobic is no air, okay? And the way you do this is you add a carbon source to the soil, you put plastic over the top, and then you add a whole lot of water. You flood it. And what happens is the microbes in the soil, they start to feed on this carbon, and they suck up all the air. It's like if you put a bunch of people into a tube, and you give them a lot of food and stuff, before long, all the air is going to be gone. And that's what you're doing. And yes, the environment would change in that tube, correct? And so that's what's happening with your anaerobic soil disinfestation. The change in the ecology, and Deirdre, I know you're talking about soil ecology. The change in the soil ecology is such that it's no longer beneficial to the pathogen. That's the principle. So you make temporary changes to the soil ecology. So that is anaerobic soil disinfestation. Are growers using it for red raspberry? They are, but they don't do the flooding. They like the benefit of putting the amendment in. There's quite a bit of nitrogen in there. They get better soil health, but no flooding. Okay, so it's being used as a kind of sort of deal. All right. So then we have mustard seed meal, which is, again, the principle is a little bit the same. You're adding an amendment to the soil at about 1 ton an acre. One of the things, though, with the mustard seed meal is that because it does generate something that can be toxic to pathogens, that's MITC, that'd be methyl isothiocyanate. It's antagonistic to the soil pathogens. And so it's a naturally derived one. So you add it in, it's about 1 ton per acre. This is some work that I did in strawberry about 2 years ago. I want to show you two things. And yeah, I don't have the laser pointer, so let me walk you through. So the graph that's on the lower left-hand side that you're looking at, do you see that there's a red line on top of the blue line, right? Do you see that? Okay, that is the methylbromide chlorpicrin standard. The red line is the mustard seed meal. The blue line is the methylbromide chlorpicrin standard. They're the same. The plants are reaching their full potential. Now look at the graph on the lower right-hand side. Do you see that? The red line is on the top and the blue line is a lot lower. What's happening? The red line, the mustard seed meal, is adding on to the deficiencies of a soil which has not been fumigated. Okay? So these are good things. I do caution you both with the ASD, the anaerobic soil disinfestation, the mustard seed meal, both of those, when you add amendments, this is what's tricky about farming. Both of those amendments are highly conducive to big populations of fusarium. The fusarium I was just talking about, it feeds on it. So you do need to know what's in your soil. If you do this in a soil with fusarium in it, you're going to get an even worse problem. So you need to know what you're doing. This soil obviously didn't have fusarium in it. So the principle of these amendments, it can make up for deficiencies of a soil to promote plant growth and performance. Okay, so now we talk a little bit about steam. This is a picture that I took. I was actually funded by the California Department of Pesticide Regulation to look at steam along with some other alternatives, and we compared them all side by side. Steam works. It's very difficult for raspberry and strawberry simply because the roots are so deep, and so you got to get it way down. So this is a picture of the rig. You have the tractor, and then the steam apparatus is drawn behind it. Do you see that cluster of people there leaning up against the fence? They weren't supposed to be there. This is private property. But you know what? When you try out something new, they come. So I got this whole peanut gallery of people looking and making comments. You know, but you try new stuff and people want to come. So we went ahead and tried it out. And what do you see? You see a lot of steam, don't you? Do you know where that steam's supposed to be? In the ground. So that's the problem. And so we do now, I didn't build the machine. It's my colleague, Dr. Stephen Fenimore. He has one for lettuce, but he only has to go 3 inches down. And so for lettuce, it works. And they have fusarium in lettuce, and fusarium in lettuce is bad news. And you put your hand on top of that soil after he steamed it. For lettuce, you can't keep your hand there. It's so hot. So that's good. Remember, you notice that I'm using the word disinfest, disinfest, disinfest. None of these techniques that we're talking about sterilize the soil. It's impossible to sterilize soil. I've gone right out after methylbromide fumigation, went and took samples. There's lots of things. You can never get everything. All you're doing are inducing shifts into the soil, which make it less amenable for the pathogen. We now have $1 million grant from the California Department of California Department of Agriculture to continue working on this steam. It's a matter of engineering. We all know steam works. It's a matter of getting the engineering right. But we need to figure something out because they're after those fumigants. We can't be complacent. We cannot. So speaking about substrate production, is there any substrate production here? Is anybody looking at it? A little bit? Okay. So it is happening now in the Watsonville, Salinas area and in the other coastal regions. The principle of this is that you're growing the plants in pots, and then you add your water and your nutrients into those pots. There are no large plantings of this, but big companies like Well-Picked or Driscoll's are actually working with this to build the circle of competence within their organizations. Because if the shoe drops on our fumigants, which it very well could at any given time, they're ready to go with the substrates. The problem with the substrates is, one, it's very expensive. I would very much like to do a cost of production study on substrates. Can't do it. There's not enough growers. So essentially a cost of production study would be a big reveal of somebody's operation. But their cost must be well above what they're bringing in so far. There is an advantage, however, with using these substrates with the caneberries, especially with the blackberries, what growers are doing, because you know those pots are portable. And you also know that California is not very cold in the wintertime. You know, it's a little hard for me to adjust. You see all these layers. I just flew in the other day. It's 75 degrees in California. So we're not getting a lot of chill, right? But we need chill to make these things grow. So what they do is take the pots, they load them, once the harvest is done, they prune them down, load them into trucks, take them into the cooler and chill them at 33 or 34 degrees for 1 to 2 months. When they come out in the spring, beautiful bud break, just beautiful, even, solid. And what happens is, you know, you always have to talk about economics. The pick speeds are extremely high because you don't have to walk very far because the fruit is so heavy on there. So rather than having a 6-week season, now you're down to a 4-week season. So there's real advantage. And yes, I have talked to the growers. Is it worth it? All this moving around stuff? Oh yeah. Oh yeah. 2 weeks where you're not paying people to do stuff is worth it. So that's another option. I will say, you know, being in around this area, I've been there since 1996. We have some of the best soils in the world on the Central Coast. We have beautiful growing conditions, the best soils, and to put pots of artificial medium, it doesn't seem right. I think we can do better, right? It just doesn't seem right. Rotation. Just a comment about rotation. We know if you rotate away from the red raspberries for X number of years, the pathogen load will go down. That's been going on for millennia. Here's the challenge on the coast of California is that the rent for land is very high. It's between $3,000 and $4,000 an acre for rent per year. Caneberries, red raspberries, about $100,000 a year crop, so it's about 3% of your cost, right? You rotate away to something like lettuce and broccoli, they're not worth that much. Lettuce is about 8,000, I think broccoli about 7,000. So if you grow lettuce on ground where you're growing raspberry, half of your cost is rent. So it's difficult to rotate. It's the economic justification of rotating away from something for so long is hard to make in our coastal region. So the berries are it, but people do know, I mean, they do know about, you can't go back to back. Back to back just increases the problem, but going 7 years, which would be the recommended, that's very difficult for us to do now. Okay, any questions on those things there? Okay, so I was going to make just a few comments on labor. And so going into this, if you take one thing home with you today, is California strawberries, because it's all fresh. California strawberries, red raspberry, and blackberry, it's all fresh. It's all picked by hand. Oh, yeah, they've tried to bring in robots, but you know, the fruit is so soft, it crushes it. The robot's not. I mean, there's 6 billion years of evolution here, people. You know, it takes a little while to build that up. So it's 60% of our costs for fresh market berries is labor. So any small shift in the price of labor makes a big difference for what we're doing. So for example, now we have Santa Barbara County, there's a push to tell the Board of Supervisors, to have the Board of Supervisors, which is 5 people that oversee the workings of the county, to push the minimum wage. The minimum wage right now is $16 an hour up to $26 an hour. Coastal California, I think it's kind of expensive here, like Bellingham. It's expensive here on the coast too, right? So the cost of living is very high on the coast. And so the workers are having difficulty paying everything. So, well, let's just pay them more. That makes a lot of sense. But the minimum wage, and I've done the cost of production studies, we have $16.50 for the payers. But if you're a grower, you also know you have costs beyond that, about 40%. You have your payroll tax, you have overhead costs, benefits. So it's about 40% higher. So $16.50 actually to the grower per hour is about $22. You raise it to $26 as what they're pushing to do. That would push it probably to about, what, $34 an hour back to the grower. You can't do it. We sent the numbers to some economists. You know, economists are very dry. And so they said over the long term, all growers would exit the industry. So we can't do that. At least not all right away. We can't do it. I mean, it's not, the facts are there. I mean, the growers are working with a very slim margin and they can't do it. I understand the travails. The work is very hard. You all know this. The work is very difficult. It's, you know, but on the other hand, the justification can't be there. And then the last comment I want to make is obviously now we're going to have a shift in federal administration. You know, there's a different attitude towards those who are undocumented in our country. The fact of the matter is in California, we have a lot of undocumented labor. So we have to see how that plays out. There are some histories with these sorts of things, see how it plays out, but I think it is only going to add to the cost of labor, some of these actions. So these are very, I'll put it this way, they're very interesting times ahead. So that does conclude my talk. I just want to emphasize again the Agdia immunostrips. Please give them a try. I think there's something there. And just so you know, I mean, there is a lot of work going on the alternatives, but it's a hard slog. There's some really smart people working on these things, and we're making very little progress. So, okay. Is there time for questions? Any? Yeah.
Have any sort of a effect?
Oh, products like bacillus biofungicides, things like that. Well, it's funny you should ask. I've done quite a bit of work with them. You know, as far as controlling diseases, absolutely not. But what is interesting is 2 years ago, together with our plant pathologist and a USDA scientist, with 2 different materials, I'll just say them, actinovate and the other was trianum, there was a significant increase in yield of the plants. And we actually had, this is funny, you know, scientists are really great. He had the fungus growing on the plant. I mean, and it established itself, a beneficial fungus that established on the plant. He didn't even share it. I called him up, hey, what's going on? And, oh yeah, it's growing on the plant, which is big. So they do that, but they don't give you prevention of disease. Some of them give enhanced plant performance. That's I've been with this since 2012 doing biologicals. It's tough. And as you know, there's like 100,000 different biologicals, right? So yeah, thanks for asking.
So our next speaker is going to be remote. And so we have time for
you.
maybe 1 or 2 questions. We're running a little behind. And then you can also text questions up here and we can give them to Mark later to answer them and he'll be around for break.
She said one more question in the back. Go ahead.
Regarding the potted raspberries, is that just something people are starting to do, or is that the direction that the
I wouldn't call that a direction. They've been doing it, you know, because in Europe they've been doing it for quite a while. I think on the Central Coast maybe about 10 years. And actually some people went in all the way. I mean, we had 50 acres of strawberries down in Santa Maria. 2-3 years later, he went bust. He ran out of money. So, you know, it's been around for a while, and by building this competence, I think is a good idea. I don't know if is that going to be the long-term solution for us, but at least they're working on it. I think hopefully I've given you the understanding. There's a lot of different angles on trying to deal with this, but fumigants are not the bet for the long-term future. No.